Hello biologists, we're going to review for the Unit 1 test for MNBA Biology, and this is Semester B. Just so you know what the test is all about, there are 15 multiple choice questions. In my class, there are 4 essay questions. Here is a practice essay question. This is not the actual essay question, but if you can answer this one, you should be able to answer the essay questions on the real test. Describe how transcription and translation work together to make proteins. The key words that you should understand and be able to use in a complete sentence are right here. If you're looking at these keywords and you don't know what some of them mean, then you probably should listen to the rest of this YouTube video and study your lessons a bit more. We'll go over all of these as we go through some practice multiple choice questions. So, which one is the best model of RNA? Look at this picture. One has a single strand and one has a double strand. If you remember correctly, RNA has a single strand, so model A is the best answer here. You notice that I've put some questions in the upside down mode over here. These are higher order thinking questions, and these are the kinds of questions that will also be covered in essay questions. So if you can answer these, you're completely ready for the essay questions. Working together, what is the role of DNA and RNA? Do they make ATP, store and use information, deliver oxygen to cells, be the main part of bones and muscles and organs, or you don't know? If you picked B, you're absolutely correct. They store and use information. Blood delivers oxygen to cells, and proteins are the main part of bones, muscles, and organs. If you want to know the answer to how is the information stored and what is the code lies in the order of the bases. It's the order of GATC or GCAU in the case of RNA that determines what order, which amino acids and in what order are lined up to make a protein. And that order of the amino acids and which amino acids are chosen makes all the proteins different. So the code lies in the bases and the order of the bases. So we see a picture here of DNA and RNA. How are they different? Why are they different? What's the structure-function relationship? Why does RNA only have one strand? Why does DNA have 2? And you can also notice they have different bases here. The difference here, thiamine is in DNA and uracil is in RNA. You don't have to know why, you just have to know that. In DNA replication, what happens? Here's a picture. We can see that this double-stranded molecule, this DNA molecule, is being split open, and 2 other double-stranded molecules are being created. And so since we know that 2 other double-stranded molecules are being created, we know that DNA is unwound and copied by enzymes. We know this is replication. This is making a copy of DNA because there are 2 double-stranded copies being created. If this was RNA, if this was transcription, only RNA would be coming out here. But this is replication because 2 double-stranded molecules are being created. The DNA is unwound and copied by these blue rectangles that represent enzymes. In DNA replication, enzymes join complementary bases. You end up with 2 complete DNA molecules. Each strand is a template for new DNA, and enzymes do unwind. The enzyme helicase here in this model is unwinding and unzipping the hydrogen bonds in the DNA. So all of the above are true. Where does protein building begin? That's a very good question. On the spindles and mitosis, on the DNA strand when RNA is transcribed, when enzymes cut up the DNA within the cell membrane, or you don't know. If you said transcription on the DNA strand here when RNA is being produced, that's the very beginning of protein building. There's only one strand in this picture because it's RNA. RNA is single-stranded. And the glob here is an enzyme. It's a polymerase, making RNA polymerase. In DNA, the correct base pair, remember DNA is GCAT, that's GCAT. So U is not correct. G and C go together. Remember GCAT. So C is the correct answer here. This process here is DNA replication, and the glob here is DNA polymerase. What is the process called when a cell uses mRNA, that's messenger RNA, transfer RNA, and the ribosomes to build proteins? That process where you take the recipe from mRNA and you make a protein is called translation. If you're looking down here, not every gene gets transcribed in every cell because not every cell needs every kind of protein that your body can make. Your heart cells or your bone cells will need very different proteins than, say, your brain. The correct sequence of events, if you look at this, is DNA is transcribed to RNA, and then RNA is translated into a protein. So if you looked at B here, DNA to RNA to protein, you're correct. B is the correct answer here. Which sequence is complementary to AGTTCA in DNA? Remember, DNA has thiamine, not uracil. So any answer with a U in it is automatically ruled out because we're talking about DNA. The answer should start with a T because we know remember GCAT, G-C-A-T, that reminds us that G goes with C and A goes with T. So this right here should be a T. So the first one should be a T. The second one is correct because G goes with C, and C is the correct answer. Which sequence is complementary to this in RNA? Now remember, there's no T in RNA. Thiamine is not an RNA. It's replaced by uracil. So here we're looking for something, we're looking for a U to start off with, then we're looking for a C, then two U's, a G, and then we're looking for a U. If you guessed C, if you guessed D, if you guessed C or B, you're incorrect. If you guessed A, congratulations. Let's look at these two pictures here. Which one is transcription? This one, or neither of them? Now remember, transcription is the process of writing down the recipe, taking that big long DNA molecule and just taking part of it and making it to RNA so you can go out to the nucleus and make a protein. So if you chose A as transcription, that's the single-stranded RNA molecule is produced, you're correct. Now we want to look for the nuclear membrane in this diagram, which is represented right here by this edge of this brown glob here. Now remember that transcription happens inside the nucleus. You can see this kind of a larger view of the cell from further away. Transcription is happening inside the nuclear membrane. Then once the mRNA is produced and kind of trimmed, it moves out to the cytoplasm and finds a ribosome. RNA contains information from a gene because the order of the bases is preserved from the DNA. Genes in the DNA are transcribed to RNA. So if you picked A, you're correct. Outside the nucleus, it all comes together. There are 3 forms of RNA working here. This is transfer RNA. It kind of looks like a T for transfer RNA. It's shaped so that it has an anticodon here and the amino acid attaches here. That anticodon matches a codon on the mRNA. This long single-stranded molecule is mRNA. And the ribosome here is all crumpled up. The ribosome is rRNA, ribosomal RNA. So there's 3 kinds right here: ribosomal RNA, messenger RNA, and transfer RNA. And here's the growing protein. After transcription, the RNA goes outside the nucleus to the cytoplasm to find a ribosome. Here's the ribosome, here's some messenger RNA. Codes on the messenger RNA that's now moved to the cytoplasm match up with that transfer RNA, and the ribosome helps the transfer RNA match up with the mRNA. And every kind of tRNA here has a different anticodon. And that anticodon determines what amino acid is going to be attached here. Remember the code, it lies in the order of the bases. Each group of 3 bases determines what amino acid is going to come in next to this protein, this polypeptide chain that's eventually going to get bigger and be a protein. And the order of the amino acids and how that protein folds up, is based on the order of the amino acids. Proteins are different because they have different amino acids in different orders and they fold differently, and that shape of the protein gives its structure and its function. The tRNA has the anticodon, and the ribosome helps match this anticodon to the codon in the messenger RNA. Remember, codons are important because they determine which amino acid is going to come in next, which amino acid and what order they come in. So tRNA and messenger RNA are going to match up. Here's the anticodon on tRNA and the codon on mRNA. Here's another view of how mRNA works with tRNA and the ribosome. The ribosome kind of supports the tRNA, hooking up here with the mRNA. And this, of course, is the amino acid chain. It's a newly formed protein. Kind of a baby protein is called a polypeptide. Which molecules determines what tRNA is used? Now remember, the anticodon, this right here on the tRNA, has to match the mRNA. So the mRNA has the code that determines what transfer RNA is next, and that determines what amino acid is next. And remember, it's the order of the amino acids and which ones are used that determine the shape of the protein, and then that shape determines its function. Look at this picture carefully and figure out what the big green thing is. If you look carefully, you can see these blue things are transfer RNAs. This ribbon type thing here is the mRNA. So I hope you guessed by now that the green thing is the ribosome. Here's another practice essay question. How do the 3 types of RNA work together to make a protein? Hopefully you can look at this diagram, identify which 3 types of RNA are there and which is which. You should also be able to identify the growing protein, the amino acid chain that's making a polypeptide. Here's a review of the codons. Here's mRNA. 3 bases makes